Hi, I'm Jennifer Simeone, and I'm a senior scientist at Waters Corporation. I specialize in bioanalytical applications for the Waters Pharmaceutical and Life Sciences team. Today, I'm going to show you how to use convergence chromatography as a separation technique to reduce matrix effects caused by phospholipids. In quantitative bioanalysis, UPLC-MS/MS is usually the technique of choice because of its high selectivity and high sensitivity. One of the challenges in bioanalysis is managing matrix effects, where components within biological samples can interfere with the mass spectrometer's response to the analyte of interest and produce erroneous results. One of the primary sources of matrix effects in plasma samples are the coelution of endogenous phospholipids. To manage matrix effects, we can focus on sample preparation techniques, we can vary the modes of MS ionization, or we can adjust chromatographic parameters. When we look at using selectivity in reverse phase chromatography, relatively hydrophobic phospholipids require high percentages of organic solvent to elute from the column. So there's a problem with hydrophobic compounds of interest, which are then likely to coelute with lipid interferences and experience matrix effects. Ultra-performance convergence chromatography, which we call UPC², uses supercritical carbon dioxide as its mobile phase, letting us take advantage of selectivity differences compared to standard reverse phase LC. The Waters Acuity UPC² system for convergence chromatography is designed to manage mixtures of compressed carbon dioxide and organic modifiers within an ultra-low dispersion system. UPC² offers those of us doing bioanalysis a very valuable orthogonal technique to manage matrix effects in certain samples. As an example, let's take a look at the analysis of clopidogrel, also known as brand name Plavix, which went off patent in 2012 and is now being produced as a generic drug. Clopidogrel is a compound that, under normal reverse phase UPLC conditions, coelutes with phospholipids. Let's compare the separation results we get with UPLC and UPC². We used protein precipitation to extract the compound from human plasma. For UPLC, we ran a reverse phase separation using an Acuity UPLC BEHC18 column. We used water and acetonitrile modified with ammonium hydroxide as the mobile phase. Then, we took the same sample and injected it onto the UPC² system using a UPC² BEH column and gradient made up of carbon dioxide and methanol with ammonium hydroxide as the additive to the methanol. Let's look at the data. In this figure, we see that the retention of clopidogrel moves from 2.41 minutes in UPLC to 0.88 minutes using UPC². With UPC², the phospholipids are clearly separated, eluting towards the end of the gradient. So for hydrophobic drugs like clopidogrel, we can effectively use UPC² as an orthogonal technique to UPLC to separate matrix effects caused by phospholipids with minimal method development time or by using advanced sample preparation techniques. Orthogonal separations, particularly useful for hydrophobic compounds, are just one of the many benefits that UPC² can bring to the bioanalytical laboratory. Learn about more applications by going to our website, waters.com/bioanalysis. I'm Jennifer Simeone for Waters Corporation. Thank you.